Good day, everyone. I will be talking about gram positive bacteria, particularly gram positive cocci. So after this lecture, the students will be able to achieve the following learning outcomes. Now, I'd like to put emphasis on second and the third um, learning goals because this topic will basically talk about the genera Staphylococci and Streptococci. So let's start first with defining what gram-positive cocci is. Let's break down terms. So again, when you say gram-positive, this would basically mean that the organism that was stained using the gram staining procedure actually took up the primary dye, which is the crystal violet. Okay? And upon decolorization, that bacteria having a thick peptidoglycan layer, if you can still recall, that thick peptidoglycan layer becomes coagulated or denatured upon decolorization. And so during adding secondary stain, which is your safranin, what happens is that the bacteria retains the crystal violet iodine complex inside, and safranin does not enter the cell. And so what happens is that this gram-positive bacteria, okay, retains color violet even upon addition of your safranin. So that's why they're considered gram-positive. And as to morphology, they are spherical in shape, no, or cocci. So in other words, you say gram-positive cocci. These are organisms which retains the primary dye during gram staining, and upon viewing the microscope, they are spherical in shape. So as a review, there is actually a test, no? A biochemical test, which is used to differentiate different gram-positive cocci. So generally, we have two major gram-positive cocci genera, that is Streptococcus and Staphylococcus. And a better test, no, a test that is used to differentiate two genera from each other, we have this what we call catalase test. If you can still recall, in doing the catalase test, we add hydrogen peroxide to the organism in question, and then the positive result is copious production of air bubbles. Okay? Copious production or active bubbling. Now, if you just say bubble production, that is actually incorrect because very few bubble production is considered negative for catalase test. Okay? So a more correct answer would be to say active bubble production or copious bubble production, active bubbling. Now, the genus giving us a negative catalase test, meaning the genus is negative because it does not contain any catalase enzyme, is your Streptococcus, whereas the genus having catalase, and so positive for catalase test, is the genus Staphylococcus aureus. Okay? Now, under Staphylococcus, we actually have a lot of species. Now, the test used to differentiate, to identify these species, to differentiate these species, is actually your coagulase test. And if you can still recall, coagulase test may be done using two methods. Okay, tube method to detect pre-coagulase, and slide method to detect your bound coagulase. Now, coagulase test, okay, a positive result for A tube coagulase test is presence of or formation of fibrin clots, whereas a positive test coagulase method is actually um, denoted by formation of agglutination. Now, the species giving us a positive coagulase test is Staphylococcus aureus, whereas those negative for these tests are generally called or classified under coagulase negative Staphylococcus or cons, no, or CONS. Now, under cons, we have um, generally two, okay? Coagulase negative Staphylococcus. Now, to differentiate these two, Staphylococcus epidermidis and Staphylococcus saprophyticus, we do novobiocin sensitivity testing and um, sensitive, okay? Sensitive to the effects of novobiocin, meaning this particular organism is inhibited by the antibiotic novobiocin, is Staphylococcus epidermidis. Resistant to the effect of novobiocin is Staphylococcus saprophyticus. We will discuss these organisms one by one. Let's start with the generalities of the genus Staphylococcus or Staphylococci. So again, as I've mentioned, cocci, no, these are spherical cells, okay, arranged in clusters. Now, when asked during examination, when shown with this gram staining result, and then when asked, okay, what is the gram staining, what is the bacterial morphology of the organism, you should answer two things. Okay, first should be the gram staining reaction. No? Is it gram positive or gram negative? Then that must be followed by the arrangement, no? The morphology of the bacteria as seen here. Now, as to morphology, okay, you do not just answer um, gram positive cocci. Has to be described what particular arrangement of the cocci is or configuration. Supposing it could be gram positive cocci in pairs, okay, if this is the one pointed under the microscope, or diplococci, or um, gram positive in clusters, okay, clusters of it, or could be in tetrads or could be in chains, as shown in the figure here. So generally, Staphylococci are non-motile and they do not form spores. So as to culture characteristics, they grow readily on most bacteriologic media under aerobic or microaerophilic conditions. So again, when you say aerobic, these organisms actually utilizes oxygen, okay, as its source for energy. And if they are aerobic, that would mean they have the enzymes not necessary to convert or to reduce superoxide radical into a harmless water and oxygen. So again, what are those enzymes? So we have peroxidase, catalase, and superoxide dismutase. Or some strains of Staphylococcus in general maybe may still grow in microaerophilic condition. So again, you say microaerophilic, no, micro meaning low amounts, aero meaning air or oxygen, philic meaning loving. So they basically could grow in conditions or environment where there is low or low oxygen tension or low amounts of oxygen. Okay, that's why they they are termed as um, they could the term is microaerophilic, okay, and these organisms, no, rapidly growing at 37 degrees centigrade produces pigment. Okay, they produce pigment at room temperature So we say room temperature, it should be 20 to 25 degrees centigrade. And this pigment is actually characteristic of Staphylococcus, no, depending on the species. Now, if it is Staphylococcus aureus, the pigment produced is staphyloxanthin, okay? Staphyloxanthin, staphylo, and then xanthin is Xanthin. Staphyloxanthin, that is basically described as a deep or golden yellow pigment. Now, if it is Staphylococcus epidermidis, okay, or Saprophyticus, the pigment produced is whitish or grayish. So as you can see here, the colonies as seen on a blood agar, no, this is actually a blood agar. The colonies that actually seen here are round, no? Round, smooth, and glistening. Now, it's actually glistening. As you can see, it glistens. This basically denotes presence of polysaccharide capsule by the organism, in the organism. Okay, this means that a colony is glistening or mucoid. That basically denotes that the organism in question or in the culture contains polysaccharide capsule. Take note of that. Now, this organism slowly ferment carbohydrates. Now, by fermentation, we actually mean that this organism is able to utilize, okay, able to utilize carbohydrates in the absence of oxygen as its source of energy. Now, there are mechanisms through which the Staphylococci genera becomes resistant to the effect of different antimicrobial drugs or antibiotics. Now, these are the following mechanisms. Number one, beta-lactamase production. Now, remember, um, generally, okay, your Staphylococcus bacteria or gram-positive bacteria having a thick peptidoglycan layer in the cell wall, okay, are susceptible to the effects of beta-lactam antibiotics. Now, in order for it to evade the effects of this beta-lactams, no, it becomes able to produce beta-lactamase, which basically breaks down the beta-lactam antibiotics. And generally, if you can still recall, okay, in the growth cell cycle, okay, of generally all gram-positive bacteria, they become susceptible in they become susceptible by the effects beta-lactam antibiotics in the logarithmic phase, because in the logarithmic phase, that is basically where cell wall synthesis happens. Okay? And therefore, production of beta-lactamase, which is able to um, resist, no, or counter the effects of beta-lactam antibiotics, okay, would mean that the organism becomes resistant to its effect. So examples of your beta-lactam antibiotics are your penicillin, okay, very common examples. Also, your penicillin-based cephalosporins. Then another mechanism is through mutation in MEC-A and MEC-C genes. Okay. Now, when organisms um, undergo mutation in these particular genes, they become resistant to these sets of antibiotics. So we have methicillin, oxacillin, and nafcillin. So this is basically the mechanism, no, of producing methicillin-resistant Staphylococcus aureus or your MRSA. Now, MRSA are generally treated no, in the clinical setting with vancomycin. However, when an organism develops mutation in the VAN-A gene, it becomes resistant to vancomycin. Now, for organisms which are resistant to vancomycin, we treat them in the clinical setting with linezolid. Okay? Linezolid. Linezolid, linezolid. Now, in general, a certain microorganism becomes resistant to the effects of antibiotics because they develop tolerance. Let's start with Staphylococcus aureus. So again, when asked what is the bacterial morphology, okay, you should answer Gram positive cocci in grape-like clusters. Okay, take note of that. Now, when shown only, a gram staining reaction with no biochemical test, you are not able to answer. You should be you should not be able to answer Staphylococcus aureus because there's actually no test done no, unless you're given with catalase positive, coagulase negative information. But when just shown with a gram staining reaction and the question says, what is the bacterial morphology? You should be answering only gram positive cocci in grape-like clusters. Okay? Now to note, your Staphylococcus aureus is beta hemolytic because it is capable of producing a factor we call alpha hemolysin. So alpha hemolysin is responsible for beta hemolysis of Staphylococcus aureus colonies in blood agar, and when this organism is incubated at room temperature, that's 20 to 25 degrees, it produces yellow or golden colonies. Okay? So this is due to production of what pigment again? Staphyloxanthin. Now to note, no? Just recall that to differentiate Staphylococcus from Streptococcus, basically, we do the catalase test. And generally, Staphylococcus aureus or the Staphylococcus genus is catalase positive. We also do coagulase test to differentiate it from cons. So, or coagulase negative Staphylococcus aureus gives us a coagulase positive reaction. Now to note, another enzyme that Staphylococcus aureus contains is this, what we call DNase. Now, your DNase is capable actually of um, destroying, no? It's actually a virulent factor of your Staphylococcus aureus capable of destroying the host or penetrating the host um, DNA. Important to note also is that your Staphylococcus aureus is halo tolerant. This basically means, no, the word halo here means salt, meaning this organism is able to tolerate, no, high amounts of salt. That's why it's also called halo tolerant, meaning it could grow. It's not inhibited by the presence of salt. Therefore, a media, no, a culture media that is used to isolate your Staphylococcus aureus should contain salt. For example, your mannitol salt agar, no, your or MSA. So it's kind of being a normal flora. It's kind of everywhere, the surface of our body, particularly the skin, no, and our nose, and we transmit this to another individual via direct contact through fomites or contaminated food. Now, as shown here, this is actually a table highlighting the different virulent virulence factors of Staphylococcus aureus. So when you say virulence, okay, virulence, that would basically mean that being virulent, no? If you have this particular factor, these factors actually, we say virulent, that means these are the factors, specific substances present in the organism which is responsible for the disease process in a particular individual, in a particular individual. Okay, so we have the following cell surface proteins. Okay, meaning these um, factor, cell surface proteins are found on the cell membrane or cell wall of the microorganism. So they help attach the microorganism to the target tissue or the host tissue. Now, I mentioned that the colonies of Staphylococcus in general are glistening. And so, glistening would actually translate to containing a polysaccharide capsule. Okay? So, this capsule is basically um, used, no, or it renders the organism resistant to the effects of phagocytosis. It's acting actually like a shield, no, like a shield protecting the microorganism from being eaten up by a phagocyte. So we have here your protein A, your Panton Valentine leucocidin or your PVL. And then I mentioned this already, no? Alpha hemolysin responsible for beta hemolysis in blood agar. Then some other factors, no, to note, a very important factor is your toxic shock syndrome, TSST1, or your exfoliative toxin. Now, this toxic shock syndrome, okay, when produced by the organism, actually makes, okay, the individual really will the individual really go morbid, no? Or undergo shock. When you say shock, no, this is basically denoted by hypotension, tachycardia, okay, or any unstable instability in the vital signs of the patient. So that is basically because of decreased perfusion of decreased perfusion in the vital organs of an individual. That's why we term it generally a shock. No? And then when you say exfoliative toxin, okay, this is basically responsible for exfoliation, no, of the epidermal layer of a particular individual. No? That's why it's called exfoliative toxin. Now, this clumping factor, okay, the clumping factor um, or the coagulase, okay, coagulase is basically responsible for clotting of the blood plasma. Now, do you need to memorize this? Yes, you have to memorize the different factors and their roles in symptomatology because this will actually give you an idea. If you see a patient in the clinics and the patient is manifesting this particular symptom, that this would actually give you an idea that the virulence factor responsible for this is this virulence factor because you knew, okay, what are its role in symptomatology or disease causation. So you have to memorize everything. Same goes with this, okay? So more or less the same table, but some additional information. So Staphylococcus aureus may actually cause disease, no, depending on the site of entry, depending where it enters the body. So when it enters the skin and soft tissues, this is associated with the following clinical conditions. When it enters the bone, infects the bone, we term it as osteomyelitis. When joints are involved, we term it as septic arthritis. Or when it gains entrance into the blood, okay? We term it as sepsis or bacteremia. Okay. In the lungs, pneumonia. In the brain, abscess meningitis. In the heart, endocarditis. In the urinary tract, infection, causing urinary tract infection. In the intestine, during food poisoning. So actually, no need to memorize this table. No, what my point here is that the disease process or the infection is actually um, based, no, to where it gains entrance or enter the body, no, or the route of entry. Basically, basing the route of entry of the microorganism. Shown here are uh, pictures of patients infected with Staphylococcus aureus. So remember I mentioned that the exfoliative toxin in Staphylococcus aureus is responsible for the exfoliation of the epidermal layer of the skin, no? As seen in scalded skin syndrome. Now, another manifestation is your bolus, okay, impetigo. You say bolus impetigo, you see bulla formation here, and then you have to differentiate it from non-bolus impetigo because non-bolus impetigo is basically caused by Streptococci genera. More commonly, Staphylococcus aureus is associated with bolus impetigo. Then when it infects the skin, okay, you see here, there's actually a punctured wound, okay, that has become infected. We term it as cellulitis. So another microorganism in the Staphylococcus uh, genera is your Staphylococcus epidermidis. Okay? Now, your Staphylococcus epidermidis appears as gram-positive cocci in clusters. And to differentiate it from Staphylococcus aureus, we perform coagulase test. Okay? So this basically means that uh, the organism does not produce the coagulase enzyme, no, free or bound coagulase or the clumping factor. Now, to differentiate it from Staphylococcus saprophyticus, okay, again, we perform novel biosyn sensitivity testing such that epidermidis, okay, this particular species is inhibited by the antibiotic novobiosyn. Now, on growth, in culture media, okay, it grows as a round colonies, smooth and glistening, no? Particularly colored whitish. No? So take note of the round colonies, smooth and glistening. The glistening there again would mean that the organism is capable of producing a polysaccharide microcapsule. So it normally is a skin flora and transmitted via direct contact and autoinfection. So it's actually a most common cause of prosthetic valve endocarditis, septic arthritis, and ventriculoperitoneal shunt infection. So again, uh, as you can, if you have noticed, okay, it basically causes infection in conditions wherein a prosthetic substance is actually inside when a prosthetic substance is actually inside the body. Okay? Why? Remember I mentioned that gram-positive bacteria, no, has actually a structure in their cell wall. We call that structure glycocalyx. Remember that glycocalyx in turn is responsible for biofilm formation. And this glycocalyx actually functions as something that really allows the bacteria to adhere to foreign substances. For example, your prosthetic substances. Okay? So they adhere to these substances, and then they form colonies there, eventually producing biofilm. So when you say ventriculoperitoneal shunts, these are actually shunts, no? Um, placed in patients with hydrocephalus So this is actually used to drain the excess fluid that is accumulating um, in the meninges of the brain. Okay, uh, from the brain going drained, the fluid is drained to the ventricles rather. The fluid is drained into the peritoneum. Then we have your Staphylococcus saprophyticus. Okay, so the same, no, it's actually a gram positive cocci in clusters. Still, it's catalase positive because it belongs to the Staphylococcus genus, and to differentiate it from a catalase positive Staphylococcus aureus. Okay, we do perform coagulase test. It's negative for the coagulase enzyme, and to differentiate it from Staphylococcus epidermidis, we perform the novobiocin testing, sensitivity testing, such that it is resistant, meaning it's not inhibited, the growth is not inhibited um, by the antibiotic novobiocin. Okay? It's non-hemolytic, meaning it does not contain the alpha hemolysin. No, it's not capable of lysing red blood cells in the blood agar, and it produces whitish colony. Okay? It's actually the second most common cause of UTI in sexually active women. Now, if you consider recall, the most common cause of UTI in women is actually Escherichia coli. No, so during history taking, for you to really consider Staphylococcus saprophyticus, you have to ask the sexual history of the patient. No? If the patient is sexually active, then you might, and then having UTI, you might as well consider Staphylococcus saprophyticus. So these are its different, okay, different virulence factors, meaning the substances capable of producing disease process, no, in an individual. So we have your SSP, AS, UAFA, SDRL, and UAFB, and surface protein F. Now, very pertinent, no, very pertinent among these, very common among these surface proteins or virulence factor is that it is able to adhere to the urogenital epithelium, able to adhere. Meaning, once it adheres there, it is now capable of producing biofilm. Now, I think you've heard of this stone formation in the kidneys, no? Or your nephrolithiasis. Remember, these stones are actually foreign. And when foreign, no, being capable of producing biofilms, they adhere also to these um, stones, no, in the kidneys or in the ureters. Okay, that's why when asked what's the common organism that produces biofilm or infection in patients with nephrolithiasis, you answer Staphylococcus saprophyticus. So to differentiate it from Staphylococcus epidermidis, actually both are gram-positive bacteria, no, in clusters. They both produce white colonies. Okay? And to differentiate the two, we perform the novobiocin sensitivity testing, to which Saprophyticus is resistant to the effects of your novobiocin, and Epidermidis is sensitive to the effects of your novobiocin. So both are actually flora, no, but they differ in location because Epidermidis is located in the skin, whereas your Saprophyticus is a flora in the genital tract and perineum. So both are pathogens, opportunistic pathogens, and they are usually, they cause acquired infection. So let's go discuss the second genus, no, which is catalase negative. We have your Streptococcus, no, Streptococcus family. Now, the Streptococcus genera are actually classified into several types depending on the following criteria. So we have colonial morphology. So when we say colonial morphology, we culture using a culture media, no? For example, an agar or a broth. And then we examine its appearance, no? The appearance of the morphology. And then we describe it, and then from there, we classify the organism. So when you say hemolytic reactions, we basically take note of its ability to lyse the red blood cells in a blood agar. So as to hemolytic, no, as to its capability of hemolysis, okay, due to the hemolysin, we basically classify it into three, no? Alpha, beta, and gamma. So when you say alpha hemolysis, that would mean it's capable of partially lysing the red blood cells. Partially lysing, remember, the red blood cell in general contains hemoglobin. And the hemolysin component of the microorganism is actually capable of degrading this hemoglobin into biliverdin. Now, in alpha hemolytic streptococci, no, what basically happens is that since it contains lesser amounts of the hemolysin, the degradation of hemoglobin stops at biliverdin. So giving us a greenish surrounding area, no? Greenish agar surrounding the colonies. Okay, that's why it's greenish because of the biliverdin, verde or green. Okay. Now in beta hemolytic streptococci, okay, it contains a lot of your hemolysin such that the hemolysin there is capable of degrading the hemoglobin completely. Okay, thus producing clearing of the agar. Now, gamma hemolysis or gamma hemolytic streptococci do not contain hemolysin, and so they are not capable of destroying or lysing the red blood cells at all. So, no clearing of the surrounding area of the agar. Now, as to serologic characteristics, now remember the cell wall of gram-positive bacteria contains different proteins. No, it's actually embedded with so many substances, no, particularly proteins. And these proteins, no, when the organism enters our circulation, enters the body, this basically induces an immune response. Why immune response? Because remember, these substances are actually antigenic. They are antigen, no? And the more antigenic they are, the more immune response and the more aggressive the immune response is. And one of these immune response by our immune system is actually through antibody production. Now, these antibodies produce specifically to the substances in the cell wall of a gram-positive microorganism are detected via serologic testing, no, or serodiagnosis. Remember? Okay, I mentioned this, no, serodiagnosis. And another thing that we do use to classify streptococci into different species is through performing biochemical reaction. Actually, a lot, no, of biochemical reactions. We will discuss this one by one in the next few slides. And through ecologic features, no, more or less the same with colonial morphology, no, needed for growth, ecologic features. So as a review, again, generally these are catalase negative, okay, meaning they do not produce the enzyme catalase. And so during upon addition with H2O2, what basically happens is that there is no bubble production at all or very few bubbles. I mentioned already that hemolysis is actually used to classify the organism into alpha, beta, and gamma hemolytic. So organisms in alpha hemolysis, we perform optokin sensitivity test. Your optokin is actually an antibiotic and being inhibited, no? Being inhibited by the optokine, we mean that the organism is sensitive to its effect. So sensitive to optokine is your Streptococcus pneumoniae, and resistant to the effects of optokine is your viridans group. Okay? Then we have beta hemolysis, no, producing complete lysis of red blood cells in the blood agar. Now, for beta hemolytic organisms, we perform bacitracin. Okay? Bacitracin sensitivity testing, and sensitive to the effects of bacitracin or being inhibited by bacitracin is your group A Streptococcus, or particularly Streptococcus pyogenes. The other hand, we have group B Streptococcus S agalactiae, resistant to the effects of bacitracin. So A, no? A for partial, P for optokine, B for beta, B for bacitracin. Take note of. Under gamma hemolysis, or those Streptococcus species which do not produce your hemolysin, okay? So these are differentiated as to growth in 6.5% NaCl. Now, if they grow in 6.5% NaCl, meaning the growth is not inhibited by salt, no, they are tolerant to the effects of salt. They are halotolerant, okay? We classify them under the enterococci. Uh, we classify them under enterococci, and non-resistant to the effects of salt. Okay, we have Streptococcus bovis. Bovis is actually a normal flora in the oral cavity of cats, dogs. So before, we term it as Enterococcus faecium, Enterococcus fecalis, but the new nomenclature would actually term these enterococci into Streptococcus, no? Faecium, Streptococcus fecalis. So gram staining reaction would produce organisms in chains, no? Gram positive cocci in chain. I hope you could appreciate the chain here. So this table, okay, these next two slides would actually summarize the entire discussion. Okay, so I mentioned that groupings may be due, or classification may be due to um, serologic testing results, no? Such that under serologic testing or group specific substance, we actually classify it into group A, B, A, C, G, among others. Now, hemolysis also may be used, and I've taught you this already, as to habitat as well, and culture, colonial morphology, or ecologic characteristics, no? And you say laboratory criteria, this refers to the biochemical test that I have mentioned a while ago. Now, for examination purposes, you should, okay, be memorizing the entire table, as this will highlight, no, the entire essence of the discussion. Anyway, we'll discuss this one by one. Okay, so another table, which is a continuation of the previous table, highlighting the different microorganisms, no, under the Streptococcus genera. Now let's start with Streptococcus pyogenes, or your group A Streptococci. So it's actually a main human pathogen associated with local or systemic invasion and post-streptococcal immunologic disorder. Now, post-streptococcal, it's because after the infection has been resolved, no, caused by Streptococcus pyogenes, it basically induces an immune response, no, producing immunologic disorders. And these post-streptococcal immunologic disorders may actually be of two. Okay, in here. So when it affects the kidney, we call it post-streptococcal glomerulonephritis, or acute post-streptococcal glomerulonephritis, or APSGN. And when it affects the heart, no, we term it as acute rheumatic fever or myocarditis. Okay, so we'll discuss this later. Now, going back, uh, as to morphology, as I've mentioned, in general, Streptococcus are gram-positive cocci in chains, and they are actually catalase. Catalase, they should be negative. No, they are actually catalase negative, being under the Streptococcus genera. Okay, and they produce large zones of beta hemolysis, as you can see here. There's actually clearing of the surrounding agar, no, the area of the agar surrounding the morphology, the colonies. Okay, you see clearing of the agar there. Now, take note that being beta hemolytic, this is usually differentiated from the other beta hemolytic microorganism through bacitracin testing. Okay? And another test, no, to identify that the organism in question is Streptococcus pyogenes is the PYR test, no, or the pyridonyl. This should be lidonyl, no, naphthylamide test. Positive for this test, no, formation of a red compound would mean that the organism is positive for PYR. It hydrolyzes your PYR. Okay, so it's a flora in the throat, the oropharynx, and it's usually transmitted through respiratory droplets. Now, as to virulence factors, it contains the following, no? Streptokinase, hyaluronidase, and DNase, with emphasis on the following. So we have your streptolysins. These are actually hemolysins, meaning these are the substances responsible for the beta hemolytic reaction when grown in a blood culture media or blood agar. Okay? Now, we have two types of streptolysin. We have streptolysin O and streptolysin S. Now, one of these streptolysins actually causes lysis of red blood cells on the surface of the agar, whereas the other streptolysin causes lysis of red blood cells below, no, below the agar. That's why when doing culture and streaking, no, when doing the culture, there is a need to stab the agar so as to cause lysis, no, to detect the presence of these hemolysins, no, to cause lysis of the red blood cells below the agar, no, the bottom portion of the agar. Then we have the erythrogenic toxin or the pyrogenic toxin, because pyro means fever, no, because it induces fever. And these super antigens are actually the ones responsible for the post-streptococcal immunologic disorder. No, the super antigens. Take note of that. So, as to serologic characteristics, remember I mentioned also that the microorganism may be classified, no, may be identified using serologic testing. When you say again, a recall, when you say serologic testing, we're actually detecting the antibodies which are produced, and these antibodies are specific to the substances or to the structure in the cell wall of the microorganism. So, antibodies specific to the streptolysin, okay, particularly O, the streptolysin O, is what we call ASO or streptolysin O. And when you detect high levels of ASO or streptolysin O, that would mean that the infection was actually pharyngitis, no? That the organism has caused infection in the oropharynx, causing oropharyngitis, pharyngitis in a patient. And but when the infection was through the skin, okay, you get to have DNase, DNase B. high levels of these titers. Titers would mean concentration. Okay, and then your streptokinase is actually an enzyme. As I've mentioned, it's an enzyme produced by your group A streptococci, and this enzyme actually has a medical application. It basically is used in the treatment of myocardial infarction because it lyses clots. Okay, so as you can see here, these are actually the diseases caused by your Streptococcus pyogenes. So take note, it also causes toxic shock. Okay, toxic shock syndrome. Remember, the organism under the Staphylococcus genus which causes toxic shock is actually your Staphylococcus aureus, and that is basically due to what toxin again? The TSST1. If you can still recall, no, go back to that table. Now, for Streptococcus, the particular virulence factor causing toxic shock syndrome is actually an exotoxin. Okay, an exotoxin. It also causes skin infection. Again, what's the titer? What's the antibody that is increased when there is skin infection caused by your streptococcus? Okay, your DNAs, no, anti-DNAs. How about for pharyngitis? Your ASO. Okay, take note of that. And another types of infection now. I'd like to put emphasis on the different post-streptococcal immunologic disorders. Okay, now upon having a um, prior infection, no? Pharyngitis, in particularly pharyngitis infection. Okay, what basically happens is that remember your microorganisms carries with it on its cell surface different proteins. Okay, different proteins, and one of these proteins is what we call M protein, no? Letter M, M protein. Now, this M protein actually has a similar structure to the proteins found in the cardiomyocytes or the cells in the heart. Got that? Now, remember that these substances in the cell wall of these microorganisms actually induces immune response. And when they induce immune response, chances are the immune system produces antibodies against, no, against these substances in the cell wall. That's the one I mentioned, no, the DNAs, the ASO, okay, the anti-streptokinase. These are the antibodies produced. And some of these antibodies are actually directed to the M protein. Now, even after the infection is resolved, but you still have these antibodies in your circulation, chances are since your cardiomyocytes have the same structure with the M protein, these antibodies attack your cardiomyocytes. No? That's why these we call this general principle of this attacking of the antibodies to the cardiomyocytes, we term it as molecular mimicry because these proteins, the M protein mimics the proteins in the cardiomyocyte. And when that happens now, these cardiomyocytes are destroyed by the antibodies, okay, producing now a disease process, no? Cardio, myocarditis, or acute rheumatic fever. So the general principle here causing acute rheumatic fever is your molecular mimicry. Now sometimes uh, these antibodies actually reacts with the antigen in question, forming immune complexes, okay? And these immune complexes becomes deposited in the kidneys now, and there in the kidneys, it induces injury, okay? Injury and inflammation causing now glomerulonephritis, okay? So, do not say no, it's just a simple sore throat, just a simple infection in the skin. if it is indeed infected with Streptococcus pyogenes, it will produce a sequelae, no, immunologic sequelae, no, destroying the kidneys and the heart. Okay, so this is what I have mentioned, that the toxic shock syndrome caused by Staphylococcus aureus is caused by the TSST1, whereas the streptococcal toxic shock syndrome caused by Streptococcus pyogenes is caused by an exotoxin. Okay, an exotoxin. So, however, we differentiate the two by this, no culture, negative culture result for Staphylococcus, negative site of infection comparing to Streptococcus. There's positive point of infection, no, either the throat, no, causing pharyngitis, or either the skin causing skin infections. And then usually we can isolate the microorganism through culture. So to differentiate the two, okay, the glomerulonephritis or your APSGN is usually follows post skin or pharyngitis infection. No? And then rheumatic fever only pharyngitis. So I mentioned this already, no? Mediated, this entire principle mediated by M protein, we term it as molecular mimicry. Whereas for APSGN, it's actually antibody antigen formation of antigen antibody complexes. Okay? So no tendency to be reactivated, can be reactivated, meaning once you are infected with it, you might be reinfected again. Okay? And then there's actually no need for chemoprophylaxis. We need chemoprophylaxis for ARF. Now, chemoprophylaxis would mean we give antibiotic to the patient. So another group of microorganism under the Streptococcus genus is your Streptococcus agalactiae or your group B Streptococci. Okay, these are also beta hemolytic, and a distinct characteristic of your group B Streptococci is a positive CAMP test. Okay, CAMP meaning Christie, Atkins, Munch, Peterson. Now, how do we do CAMP test? Okay, we streak Staphylococcus aureus in the center of the agar, and then another perpendicular to it, we streak a colony of group B Streptococcus. Now, you incubate this after for 24 hours to in 37 degrees centigrade. You get to see arrowhead beta hemolysis. Okay, the hemolysis is forming an arrow. That is a positive CAMP test. A negative is no formation, of course, of arrowhead hemolysis. Another test is that it's also positive for sodium hippurate, meaning it hydrolyzes sodium hippurate, the substrate sodium hippurate, okay? And it's actually a normal vaginal flora and also in the lower gastrointestinal tract. Now, to note, no, being a normal flora in the vaginal tract, what's the significance? The medical application for this is that any pregnant woman who has, okay, who has in there, no, the vaginal flora, the perineum being colonized by group B streptococci must be given with antibiotics. That antibiotics, no, at prior to giving birth, because again, your group B streptococci will actually cause neonatal infection to the newborn infant. Okay, here. So it causes urinary tract infection in a pregnant woman, but those pregnant women having had a history of urinary tract infection, what we usually do in the clinical setting is we screen for GBS colonization. So we do a swab in the perineum, in the general flora, and then this is usually done at 35 to 37 weeks age of gestation. And when it is positive for GBS colonization, what we usually do is we give chemoprophylaxis. No, we give antibiotics to the patient, to the mother, because no, we've had a lot of patients, no, pregnant women having had a history of UTI, and they did not discussed to us that they had UTI, what happened to their infant was they were actually the result was actually very morbid. No? The infant has had pneumonia, sepsis, and meningitis. Okay? It predisposes the infant into pneumonia, sepsis, and meningitis, or infection, in other words. Then we have your group B Streptococci. These are non-hemolytic and PYR negative, no, as compared to your Streptococcus pyogenes, which is PYR positive. Okay, it hydrolyzes uh, isculin and grows in the presence of bile, so it's bile isculin positive. However, it does not grow in 6.5% NaCl and is optokin sensitive, resistant rather. It also causes UTI, biliary tract infection, and endocarditis, no, marantic endocarditis in patients with abdominal malignancy due to Streptococcus bovis. Then we have, second to the last organism, we have Streptococcus pneumoniae, which is a gram-positive diplococci. Okay, so it possesses a capsule. Now, very important thing to note in Streptococcus pneumoniae is that it produces autolysins. And it's kind of difficult to culture these microorganisms because the autolysin, okay, the autolysin actually causes the microorganism to kill or lyse itself. Meaning, for you to really do, no, isolate this and have a stock culture of Streptococcus pneumoniae, you'll have to repeat, no? You'll have to do subculture as often as possible. It could also cause pneumonia, sinusitis, otitis, bronchitis, bacteremia, and some other infectious processes. Okay, so these are alpha hemolytic, as you can see here, meaning alpha, it is not capable of completely lysing your red blood cells, and that incomplete lysis of your red blood cells produces what product again? Biliverdin, okay? Thus the greenish discoloration here surrounding the colonies, okay? And to note, the growth is basically enhanced by 5 to 10% carbon dioxide. In other words, if they are carbon dioxide loving, they are capnophilic, no, capnophilic, C-A-P-N-O, capnophilic. Capno means carbon dioxide loving, okay? Such that, how do we attain the 5 to 10% CO2 conditions in their growth? No, we basically incubate the blood agar in a candle jar. No? So you've learned in basic science that for us to produce fire, we actually needed oxygen. Now we close the jar, no, with a lid. Now once all oxygen is consumed here inside the jar, no, by the fire formation, what remains is a carbon dioxide. And that carbon dioxide is utilized by your Streptococci, no, your capnophilic organisms for its growth. No? Very smart thinking, no, yet very practical. So we usually do this, no, all culture media with blood, no, blood agar culture media, chocolate agar, no. We usually incubate it under candle jar. So colonies are round, as you can see, but it forms a central depression. So you see a depression in the center. It's usually umbilicated, no, in the center, and the colonies producing polysaccharide capsule are glistening. So you see the glistening here, okay, candle jar there, and these are the different virulence factors of your Streptococcus pneumoniae, meaning these are the factors which are responsible for the disease process caused by your Streptococcus pneumoniae. So you have to memorize this. No? Particularly, we have the pneumolysin, which causes the beta hemolysis, and some other factors that you need to memorize. And as I've mentioned, your Streptococcus pneumoniae contains capsule. And this capsule is usually detected by the key-lung reaction. Okay? The key-lung reaction or the capsular swelling reaction actually will detect the presence of capsule. When there is clearing around the cell or capsular swelling that is basically positive for key-lung reaction, that means that the organism in question contains capsule. So key-lung positive Streptococcus pneumoniae. These are the diseases caused by your Streptococcus pneumoniae. I've mentioned this already, no? Being a normal flora in the respiratory tract, it can actually cause pneumonia, emphysema. In the blood, it causes septicemia, no? In the ears, it causes otitis. In the maxillary cavity, it causes sinusitis. In the peritoneum, it causes peritonitis. It gains entry into it enters the joint, it causes osteomyelitis or bones, no? In bones, osteomyelitis. And when in the meninges, it causes meningitis. So a good way to prevent infection caused by Streptococcus pneumoniae is through vaccine administration. And to note, okay, we actually have different types of vaccine that will target your Streptococcus pneumoniae, particularly your polyvalent polysaccharide vaccine. This vaccine actually has a 23 isotypes, meaning it could target the 23 types of polysaccharide capsule in the Streptococcus pneumoniae. Okay? Some vaccine are conjugated and are coupled with a carrier protein, and these vaccines are usually given to high-risk individuals. So elderly asplenic, meaning this is an individual in which the spleen has been removed. Okay, diabetic patients and patients with sickle cell disease. Then lastly, we have your Viridans streptococci. Okay, these are the representative microorganisms. These are usually uh, habitants. Okay, they inhabit the mouth, no, the oropharynx. Okay, and they are usually gram-positive cocci in change and are catalase negative and are alpha hemolytic. No, they are resistant to the effects of octokine and they are bile insoluble. So as to habitat, okay, I mentioned already that they inhabit the oropharynx and they usually cause infection when they enter the bloodstream during dental procedures. Being inhabitant of your oropharynx, okay, they adhere, no? They adhere to the surfaces there due to glycocalyx. And when it enters the bloodstream, if you have damaged valves, no, they could actually form biofilms there. They adhere there and form biofilms, no, particularly in vegetation. So S mutans causes dental caries. The one causing infection in the valves is your S sanguis, and your S intermedius causes brain abscess. So as a review, okay, as a summary of the entire discussion, so we have a catalase test, no? Take note that catalase will differentiate staph from strep. Catalase positive is your staph, and used to differentiate the different species under staph is your coagulase producing positive result, your S aureus, and your S epidermidis and saprophyticus negative for coagulase. Now, in strep, we usually use hemolysis patterns to classify it into alpha, beta, and gamma. So we have beta hemolytic pattern. We use beta for bacitracin. Sensitive to the effects of your bacitracin is your pyogenes, and CAMP test positive and Hippolyte test positive is your agalactane. Then under alpha is your, we use optokine test, no, to to classify it as optokine sensitive or resistant. No, sensitive to the effects for optokine is your strep pneumoniae, and resistant to its effect is your viridans group. Then we have your gamma hemolysis. Lastly, we usually use bile isculin, and 6.5% NaCl, such that um, positive growth in 6.5 NaCl, we classify it as enterococcus, and negative negative growth, meaning it's inhibited by 6.5 NaCl, are your non-enterococcus, particularly Strep bovis. That ends our lecture for today. Thank you very much for your kind attention, and have a good day.